This video is the second part of the breast cancer laboratory, in which I will discuss the individual slides and identify the structures you should recognize. Keep in mind that a carcinoma generally represents the proliferation of only one cell type, typically either ductal or lobular cells, but not the myoepithelial cells. Invasive ductal carcinoma represents the vast majority of breast cancers. Lobular carcinomas are far less frequent. Place a slide labeled breast 1 on a sheet of white paper and examine it by eye. Observe that the section contains clear regions of fatty tissue interspersed with dark streaks of eosinophilic staining collagen fibers. Near the center of the sample, note the presence of small cysts. Examine the slide under the 4X objective lens and locate one of the darker staining regions. You will observe that it is composed of collagenous connective tissue, while the surrounding light staining region is composed of adipose tissue. Locate a normal terminal ductal unit and switch to higher power. Examine the normal terminal duct lobule unit that is circumscribed by connective tissue. The glandular portion is composed of small acini that are lined by two layers of cells, an inner layer of lobular cells and an outer myoepithelial cells. These are flat and located on, not in, the basement membrane. Also locate ducts that are present in the collagenous connective tissue. These consist of inner ductal cells that are often multi-layered and outer myoepithelial cells. This pattern is maintained throughout the entire ductal system. Now return to low power and locate the microcysts that are in the center of the section. Adjacent to these microcysts are ducts in which the epithelium is growing into the lumen. These are identified as usual ductal hyperplasia. Examine the region between the cysts and the hyperplastic ducts under higher magnification. The upper left-hand side of the slide shows that the cyst is lined by flattened epithelial cells, and there is no indication of proliferation. On the lower right-hand side of the slide is a duct showing usual hyperplasia. Usual ductal hyperplasia consists of a haphazard growth of both ductal cells and myoepithelial cells that overlap with each other. Look for the presence of flattened nuclei that may be from myoepithelial cells. You should note that with H&E staining, it is difficult to identify myoepithelial cells. However, immunohistological staining readily reveals the presence of these cells. Also note that these cells are forming lumens that resemble normal ducts. The important thing to note here is that these cells have a variety of shapes and sizes similar to that of the normal ductal epithelium. Now examine the slide labeled breast 2. As always, first place the slide on a white piece of paper and observe that this specimen contains a large fraction of collagenous tissue. On one side of the section, you should observe some black ink. Indeed, as you go through these labs, you will encounter a number of sections that have ink on them. This deserves some explanation. This ink was applied to the outside of the specimen immediately after surgery to indicate the margins. Now, one of the important duties of the pathologist is to determine if all of the tumor cells have been removed during surgery. If the pathologist examines a specimen under the microscope and finds tumor cells close to the ink stain margin, then there is a good chance that tumor cells still remain in the patient. This is not a good thing. Examine the slide with the 4X objective lens and locate a region near the inked margin, similar to what is shown here. This view shows both a normal ductal lobular unit and an example of ductal carcinoma in situ, or DCIS. It is very easy to distinguish between these two regions. In particular, the normal lobular unit contains a large number of small ducts and acini, each of which have empty lumens. In contrast, the DCIS is extremely large and the lumen is completely filled with cells. The fact that the DCIS is close to the inked margin is not a good sign. Now switch to higher power and examine the normal lobular unit first. The normal lobular unit shown here consists of an extended terminal duct and round secretory acini. Look closely at the pattern of the basophilic nuclei in the terminal duct and note that the epithelium is either 1 or 2 cells layer thick. This is normal. Now move the slide to the region of the DCIS. Compared to the previous slide at the same magnification, the differences are readily apparent. The DCIS is much larger and filled with cells. This can be distinguished from hyperplasia by the fact that the cells look very uniform in size and shape based upon the morphology of the nuclei. Scan the slide at low power and locate other regions of DCIS. In some cases, you will find that the middle region has undergone necrosis. That is, the cells have died. As the tumor cells grow and fill the duct, some of the cells in the center become distant from the blood supply and consequently die. That is why you do not see nuclei in this region. They have been replaced by an eosinophilic substance that is hard and often shatters during sectioning. Next, examine the slide labeled breast number 3 by eye and observe that it contains several strands of collagen embedded in a matrix of adipose tissue. Now, this section contains a number of conditions and should be studied in detail. Please make a mental note of the location of the different structures indicated here. Under low power, locate the region containing the ductal carcinoma in situ, or DCIS. This DCIS is of the comedo subtype based upon the following features. First, the tumor cells have formed a solid sheet. Secondly, the tumor cells are of a high grade, in other words, not well differentiated. And third, you will find the presence of central necrosis, which I will show you next. This slide shows examples of central necrosis that is often present in the comedo subtype of DCIS. This is a higher power view of DCIS containing an eosinophilic staining necrotic region and a central basophilic region of calcification. This calcified region is hard and has been shattered in the process of sectioning. Now, calcification can occur all over the body as part of the healing process. Calcification of the breast can be easily seen on mammograms and serves as a marker for DCIS. Switch back to the 4X objective lens and locate the region of invasive ductal carcinoma. This consists of irregularly shaped tubules that merge into one another. This is a higher power view of the invasive ductal carcinoma. This would be classified as well differentiated based on the modified Bloom and Richardson grading system. Also note the presence of clusters of lymphocytes present around the tumor cells, indicating that the body is mounting an immune response to the tumor. Again, switch back to the 4X objective lens and locate the region of scar tissue, which probably resulted from a previous biopsy. Examine this region under higher power and locate a collection of giant cells, which, as the name implies, are very large and multinucleated. These cells are a type of macrophage that have converged in this region and fused together. This is a normal part of the healing process from a wound. By eye, examine breast slide number 4 and observe the presence of eosinophilic collagenous tissue that radiate out from a central region. This represents regions of wound healing. Under low power, locate the DCIS that is similar to that of the previous slides. Also locate a region of scar tissue that presumably resulted from a previous biopsy. It appears that in this patient, a biopsy had been taken, but the tumor tissue had not been completely removed and had to be re-excised. Examine the central region of scarring and identify the giant cells. Now, the fact that a DCIS was present around this biopsy cavity suggests that the original lesion had not been completely excised. By eye, observe that the slide breast number 5 is very basophilic, suggesting a high cellular content. In fact, it consists predominantly of DCIS and invasive ductal carcinoma. Note that the tumor is located fairly distant from the inked margin, suggesting that it was adequately excised by the surgeon. Under low power, observe that the bulk of this specimen consists of invasive ductal carcinoma. The tumor cells appear in various forms, as tubules, in sheets, and as single cells. Observe also the numerous DCIS at the periphery. This figure shows a higher power view of the carcinoma cells penetrating into the adipose tissue. Now, this final image is again a high power view of invasive ductal carcinoma. This tumor would be graded as moderately differentiated according to the modified Bloom and Richardson system. This is based on the following 3 criteria. First, nuclear grade. It receives a high score of 3 from very large size and prominent nucleoli. Now, I realize that the morphology of this section is not all that good, but I think you can begin to make out the size of the nuclei. Secondly, mitotic activity. Here it receives a low score of 1. Now, to get this score, you must count the number of mitotic figures over 40 high-power fields. So for this, you just have to take my word for it. And 3, tubule formation. Here it receives a middle score of 2, in which 20 to 40% of the cells have formed tubules. In this particular view, I don't see any tubule formation whatsoever, but in other regions there is some. So when you add all 3 scores together, you get a total of 6, and that indicates a moderately differentiated invasive ductal carcinoma.